Okay, this is the first in a series of videos about gene expression. The first video is going to cover the first section in our class notes on gene expression number 1, where we're looking at defining the terms genome and gene and understanding what we mean by coding or DNA and non-coding DNA, functional RNA, and basically the structure of DNA and RNA. So those first few little points there, 4.1.3.1 and 4.1.3.2 is what we're covering now. So this is what we're covering first. Let's see, we've got our learning goals there, but we're only going to cover a few of them right now. We're covering this one here and this one here in this video. So, purpose of DNA. First of all, DNA is essentially what encodes for our genetic recipes. Now, these are the recipes that we use to make functional molecules. So these are little molecules that actually do jobs in the cell and allow our cell to stay alive. From structural molecules to ones that actually are little machines, all of them have their own genetic recipes, and those recipes are found in the DNA. So some examples of this is simply when you have an organism that has enzymes. So enzymes are machines or little protein machines that actually catalyze our reactions and speed up the reactions so that they get done fast enough for the cell to survive. Enzymes will easily speed up reactions by something like a factor of 10,000 times a second. So they're fantastic for speeding up cellular reactions. You've also got hormones and signaling molecules. So those little guys help us to interact and react to the environment around us, which is really important. And the other thing that we do is we have molecules that allow the cell to reproduce itself, to clone itself, to repair itself. Now, each one of these recipes, they outline basically the ingredients for the functional molecule and the order in which those ingredients need to be put together. That's essentially what a gene does. The ingredients and the order in which they're put together. And those two factors, along with the place in the cell that they're made, actually allow them to fold up in a way that makes them a functional molecule. Next of note in your notes is that I've made you realize that when I say recipe, I actually mean a gene or a piece of DNA that codes for a functional molecule. Okay, so that's essentially what a gene is, a piece of DNA that encodes for a functional molecule, and it doesn't have to be protein, it could actually be RNA. We've also got a little point there that says importance of order and type. It affects the folding and the structure. We can go into that a little bit in extension, but essentially, let's say for a protein, if you have a protein that's made up of sections of hydrophilic or hydrophobic amino acids. So if there's, say, a lot of hydrophobic amino acids, those amino acids hate water. But we pump out that chain of amino acids into the cytoplasm, which is a water-filled environment. So as soon as they start coming out, those amino acids are going to start clumping in on each other and folding, because their very nature, the fact that they're hydrophobic, will mean that they will fold as soon as they enter a water-filled environment. So that's what I mean in your notes when I said there is an importance of order and type, because when you get proteins like this that have structures like this, protein structures like this, that's actually a structure that's developed by having sections of hydrophobic and hydrophilic, water-loving sections that will then clump together and then turn and then clump together and turn by having that alternating water-loving and water-hating sections. So the order of the amino acids, or the order of the nucleotides for RNA, and their characteristics, so whether they're water-loving, water-hating, what type of charge they have, that actually will then affect how they fold when they're made. That's what that point's about. Now, we've also got here something sections of DNA are equally important to coding DNA. That is, non-coding sections of DNA are equally important to coding DNA. We used to call them junk, but they're not. Okay, the non-coding sections of DNA actually help turn genes on and off. They react to our environment. They actually allow us to transfer traits or silence traits even down into our kids. They even allow us to pass information like we know that we can actually pass genetic information that will prepare our babies' bodies for a famine. If the mother or the father are exposed to famines at certain times in their life, it will actually affect the code that goes into the sperm and eggs that will then affect the children that they have. So the non-coding sections are extremely important. There is no such thing as junk DNA. They support several things. They might support the DNA structure, so keep it safe and keep it stored well. They can encode for functional RNAs. They can be removed later on. We call that spliced during transcription, but they remove different ways to produce different types of protein. They're incredibly important. I can't say that enough. Anyway, let's go on to the next. So what is the basic structure of DNA? Okay, so the first thing you've got to realize is that the DNA essentially has two sections, coding and non-coding. Okay, I think I have a picture somewhere down below here. I'll just move up a little bit here. There it is. So DNA has several sections. You have coding sections and non-coding sections. Where you have a coding section, you have a gene. So this thing here codes for something. And we have non-coding sections like over here. And those little guys, they actually don't code for any proteins, but they are extremely important. You see, for a gene to actually be turned into a protein or a functional RNA, first you've got to actually get the little molecules, polymerase and stuff, that are the ones that actually copy it. You've actually got to attract them to the right place and show them where to actually lock on and tell them, yes, you need to copy this little piece of DNA, and oh, you need to stop here. Like, it's actually the areas either side of the gene that determine whether or not this little section here gets copied at all. Without those areas on either side of the gene, or even well down from the gene, like down here, you will actually have that gene essentially just sitting there doing nothing. So it's really important that we actually, I'm just changing this around, it's really important that we realize that those non-coding sections are extremely important as well. So let's go over a few of these things. Just going to tap back here. Okay, so in the non-coding DNA, we have areas where they are structural proteins, okay, structural pieces of DNA. So these guys basically support the DNA, but they don't actually get transcribed or photocopied into RNA. We have areas which then, when they are transcribed, they actually form functional RNA. So RNA machines like ribosomes, messenger RNA, there's actually heaps of them, microRNAs, heaps of these tiny little RNA molecules that we never knew did anything, that are actually incredibly important for turning genes on and off. And if it wasn't for those functional RNAs, nothing would work the way it does. We're only just touching the surface of that one. Introns, they're pieces of the transcribed DNA, so the photocopied DNA, that for some reason they remove after the fact. And you think, well, if you're going to transcribe or make a photocopy of DNA, and then you're going to cancel out all these areas here, why the heck didn't you just cancel that out to begin with? Well, we realized that one section of a gene, like this, I'll go to the next slide and that might help, just a minute. One section of a gene like this, you've got several little sections where you've got these introns, the things that are going to be cut out before we send it out to become a protein. These little guys get cut out. However, they don't always get cut out. Some of them get left in. So this one little gene here, it can sometimes, and I'll scribble over this in a different color if I can. Let's try this one. Sometimes you'll transcribe or copy this whole section here, and just this bit will get spliced out, which means you'll have a shorter gene that will then go and make a certain protein. Sometimes you'll cut out two pieces, and that will make a different type of protein based on the same recipe. It's sort of like having a cake, and sometimes you have chocolate and sometimes you have chocolate and and caramel or whatever. So it's using this one gene recipe to make many different types of proteins, if that makes sense. So we realize later on that the introns are really important because they either get taken out or left in depending on the variation of the protein that we are making. So one gene makes many different proteins. So, and again, we're only just starting to figure this out now. So let's go back and have a look at those notes again. So that's introns. And last but not least, we have this other non-coding DNA. We don't know what all of it does. We are only just starting to scratch the surface, but under no circumstances are we ever going to call it junk DNA ever again. We really got a shock when we realized all that junk DNA wasn't junk at all. So we're pretty sure it has some job to do, but we've not put enough time and effort into actually trying to find out what it does yet. So that is actually the forefront of genetic science at this point in time, specifically in how some of that non-coding DNA turns into RNAs, which then go and affect everything else. So that's the end of this video. We're going to go on to the next video, which is going to be looking at how DNA is stored in the cell. Before we just go on, I'm just going to point out a few things when it comes to this DNA structure here so you can understand it. You've got your chromosome. You've got that chromosome is actually long ropes of DNA. And we're going to be learning soon about that DNA is actually coiled around these little proteins called nucleosomes here, which actually store the DNA. But when you come to a gene section here, you always have a gene. You have a gene like that. And you always have sections at the front and at the back that help show the RNA polymerase and all of the transcription factors that help point the body to what part of that DNA needs to be photocopied. You have these little guys that actually influence whether or not that gene actually gets transcribed at all. If these guys aren't turned on, nothing happens. There are 4 meters of DNA in every cell and something like 10,000 plus genes. So it's only if these little areas here are turned on that gene actually then gets copied and turned into either mRNA or functional RNA. So in those little areas there we call them a promoter region there. Don't worry about the rest. You don't need to know all of these. There's also another area here that's well upstream of it called an enhancer region or a silencer region. If he's not turned on and happy, nothing else happens. And at the very end here, you have other areas here which we call regulatory sequences again, and they have a stop codon there that actually tells the RNA polymerase, hey, you've finished copying the gene, you can stop now. And there's other areas down below here which will again affect whether or not anything can attach at all to copy that gene. So it's quite a complex situation that you have to get your head around. You need to know the basic structure, but all of this stuff you don't need to know specifically for the test.